Good day everyone, I'm Jun Yao, and today I'm going to share an interesting article which applies the knowledge that I have learned from the course SID3016 Applied Chemometrics. The article that I have chosen is Production of Biodiesel from Crude Palm Oil by a sequential hydrolysis and esterification process using sulfuric water. The link to this article can be accessed through this QR code. Before we move on to the main part, let me do some brief introduction on the production technology of palm biodiesel first. So basically, crude palm oil consists of mainly triglycerides along with trace amount of diglycerides and monoglycerides as well as the free fatty acids. The conventional way of producing biodiesel from palm oil is by trans-esterification, where the triglycerides in the crude palm oil are trans-esterified to form fatty acid methyl esters, which is basically our biodiesel. So for the article that I am sharing today, it provides an alternative to the production of biodiesel by the combination of hydrolysis and esterification, which is known as hydroesterification. In this article, the knowledge on the chemometrics is applied in the first part of the reaction, which is hydrolysis. This reaction is investigated by using a 2 to the power of 4 full factorial design. In this design, there are 4 variables being studied, which are water to oil molar ratio, temperature, reaction time, and stirrer. For each variable, there are values and their respective coded levels, which are illustrated in this table. The response factor in this design is the percentage of free fatty acids content. With all the 4 variables presented, the full factorial model can be constructed and it is represented by the following equation here, where y is the response factor, x are the 4 different variables, beta are the constants or the coefficient in this case, and epsilon is the random error term. In this article, JMP and MATLAB were used to calculate the coefficients of the model. Furthermore, there are 5 extra replicates being done at the center point added to estimate the experimental error, the data reproducibility, and the potential curvature. The model was evaluated by analysis of variance, also known as ANOVA, at the 95% level of significance using F-test and T-test. So the 4 variables studied along with the results are summarized in this table. The conversion of triglycerides into free fatty acids is from 9.9% to 86.4% depending on the conditions of the reaction. Various tests was carried out also to determine the significance of each variable in the design model. For one variable or even the interaction to be significant to convert triglycerides into free fatty acids, the p-value must be lower than 0.05. The result of this variance test is illustrated in the table as well. From this table, we can determine that the temperature and the reaction time play the crucial role in the conversion reaction, where the former is more important than the latter. Meanwhile, since the other variables and interactions present p-value much higher than 0.05, we can conclude that they do not possess significant role in the conversion reaction. In the ANOVA, which is shown here, the variation between sample means of the model regression was much greater than tabular f-value for the same degrees of freedom of 3 sources of variance. Furthermore, by observing the f-value of lack of fit, which is 0.007, we can conclude that the established regression adequately represents the data, the model has good ability to estimate the response, and also the lack of fit of the model was not significant relative to the pure error. Based on the statistical analysis of the results, an empirical model can be constructed to describe the variation in the free fatty acid concentration as a function of temperature, time, and the temperature quadratic term. The last term is a representative of the presence of curvature in the model, and this was actually confirmed with the curvature test. Based on the analysis, temperature is a crucial factor to the yield of hydrolysis reaction, and this situation can also be explained by using the chemistry theory as well. High temperature decreases the dielectric constant of the water, and this makes the dissolution properties of the subcritical water similar to the oils, increasing the interaction of oil in water as well as the rate of reaction. Other than temperature, the reaction time is also important in providing high free fatty acids content. Theoretically, the hydrolysis of triglycerides occurs in 3 stepwise, where the triglycerides can be converted into diglycerides, then monoglycerides and glycerol, and each step will produce free fatty acids. Thus, the longer the reaction time, the more complete the stepwise reaction will be, and the higher the content of the free fatty acids. So these are basically where the knowledge of chemometrics is used in study and experiment to determine the significance of each variable in the experiment, which can be supported by the theory that we have learned in chemistry. Thank you for the attention.